It's been another 14 days, folks. Your extended family has circled. It's so nice to see all of you this evening. And for whatever reason you are here, we are all here for you. So you're never alone. And it feels really good to see all of your faces. So thank you for making our extended family cozy. And welcome to a new program from Everything ALS to all of you into your living room.
Good evening or good afternoon, depending on where you're at. Thank you, McFinn, for kicking us off. And I am so glad that you're all here with us tonight. I'm super excited to introduce our guest speakers tonight. We have Dr. Alan Cox. He is the executive director at the ethnobotany and drug discovery at the brain chemistry labs. And with him is Dr. Sandra Bannock, and she is their senior scientist of ethnobotany. So Dr. Cox has spent over 30 years living in small villages, working on drug discovery programs and doing fascinating research. And his research has been focused on neurodegenerative illness with the goal of discovering new therapies for ALS. And Dr. Cox is super patient-centric, and he has a ton of compassion, and he understands the challenges that people with ALS face. And he knows that time is of the essence and proven therapies are needed now. So Dr. Cox has also published over 200 scientific papers for books. He was in the film Toxic Puzzle, which was a very interesting medical and environmental detective story about environmental toxins. Time magazine had named him one of the heroes of medicine, and for his work in ethnobotanical drug discovery. That's a tongue twister for me. So thank you for being here tonight, Dr. Cox and Dr. Bannock. The floor is all yours.
Thank you. It's great to be with you. A short introduction of Sandra. She is our senior scientist. She took her Ph.D. at the University of California, Berkeley, and gave up lifetime tenure in the California state system to come work with us in Jackson Hole to try work on ALS. Let me go to my screen here. We're gonna hopefully go about 35 minutes and then we'll stop for questions and discussion. I've been working most of my career on drug discovery. I was trained as in evolutionary biology. I took my PhD at Harvard. There, a very famous ethnobotanist discovering that I could speak Polynesian languages because I had served as a missionary for the Church of Jesus Christ Latter-day Saints. Richard Evans Schultes was his name. He said, oh, you know, you need to do ethnobotany. So I did it in the field work in the islands, sort of as a secondary endeavor. But then when my mother became ill with metastatic breast cancer and died of it, I decided to pivot and focus my major efforts on drug discovery. I played a small role in the discovery and advancement of Taxol, which is one of the major drugs now used for the treatment of ovarian and breast cancer. In my laboratory, we discovered 5 different drug candidates for different things like immunostimulation, inflammation. But in the 80s and 90s, I'd discovered from studies of indigenous healers in Samoa, a new candidate for the treatment of HIV/AIDS called prostratin. Once that got launched, I wanted to find a new disease indication to work on that had sort of a sense of hopelessness and that there hadn't been many drugs discovered, one that took out whole families, and settled on ALS. Realizing that the physicians, neurologists had ALS since its characterization by the French neurologist Charcot for maybe 150 years or so, and there were no really good drugs coming out. I thought, well, this is going to require more than just neurology. So I recruited a very diverse group of scientists. We settled in Jackson Hole for some reasons, become obvious, and we devote our full-time research to defeating this and other progressive neurodegenerative diseases. At the same time, in the 90s, late 90s, there was a real explosion in the understanding of genetics and risk factors. But unfortunately, this is not translated into new pharmaceuticals that patients can take. And you know, in retrospect, it's sort of obvious. We know about 10% of ALS patients have a familial history of disease, but 90% is sporadic. Most people, when they get the diagnosis, you know, have actually not heard of the disease before. And what something we'll come to in the end, Dr. Bannock come to the end, is we're very concerned about the time it takes patients to receive a firm diagnosis. And we've set that as a target to try speed up diagnostic procedures. So we created a not-for-profit organization called the Brain Chemistry Labs in Jackson Hole. People sometimes say, well, why don't you make a biotech? And the answer is because, you know, we can move so much faster as a not-for-profit. We don't have the profit motive out there. All we care about here is changing patient outcomes. And we're able to do experiments that probably wouldn't be viable in a pharmaceutical setting because they're always worried about sunk costs and, you know, profitability. We don't care. We just want to change patient outcomes for ALS patients. So we're founded as a not-for-profit organization in Jackson, Wyoming. Our total focus, our total aim is to improve patient outcomes. We have no other reason for existence. We are very focused on stopping this disease and on preventing it. And we recruit the top minds we can find, realizing that it would require an international and interdisciplinary effort to really get a serious bite into ALS. I went around the world and gave investigators what I call my John Galt speech, which is, gee, you're a big deal. You've published books, you've won ribbons, you've got an endowed chair. So I want you to set that all aside now and come work with us in Wyoming to focus on this disease. And out of those 50 pitches, every single person I've asked has agreed to do that. So we anchor a small team of 5 PhDs in Jackson, but we have 28 different laboratories around the world that we work with on a weekly and sometimes daily basis. Our focus also is to move very quickly to clinical trials. While we respect those people are, you know, focused on mechanisms of action, we aren't. We want to get drugs out fast that can help ALS patients. Let's see here. So we get our consortium together once a year, all together. We've had meetings in Johannesburg and in Stockholm and here in Jackson Hole. We've recruited some of the world's top neurologists and ALS experts. You might recognize Dr. Walter Bradley, Chairman Emeritus of the Miller School of Medicine, Elijah Stommel, Dartmouth Medicine, John Weiss, University of California, Irvine Neurology. We also have oceanographers, we have synthetic chemists, we have microbiologists, a lot of different people from all over the world, all focused on this single goal of trying to come together to figure out a way to take out this disease. And so I guess the first message I really want to leave with families and patients is that take some hope here. You know, every morning we wake up in Wyoming, we're thinking about ALS, and we've got a massive team of really world-class scientists that are helping us push the research forward. We decided, I'm an ethnobotanist by training. I'm not a neurologist. I'm not a physician. I'm an ethnobotanist. So I and a small team began our work by studying a disease suffered by the Chamorro people of Guam. This is an island in the Pacific. I'd recruited Susan Murch on the screen here. She had run amino acid analysis at Sick Children's Hospital in Toronto, probably the top amino acid chemist in the world. She now holds a chair at the University of British Columbia. Here's Dr. Bannock, superb analytical chemist in her own right, and an expert on plant-animal interactions. We had some physicians, Dr. Patricia Stewart, and some neurologists, Dr. John Steele, others that came with us. We started very quietly making field trips to Guam to try to figure out this disease. Here I am in the Umatic village graveyard. There were two villages, remote villages in Guam, that a quarter of the people died of a very puzzling paralytic disease that had symptoms similar to ALS, Parkinson's, and Alzheimer's disease. Our thinking was that if we could figure out what was killing these villagers, that we could not only help them, but it might give us some deeper insights into ALS. So we came in a very orthogonal path towards ALS therapy by going to small villages. I think we've been in every village in the world that has an elevated case of ALS, incidence of ALS. Marjorie Whiting, who had been at the Burns School of Medicine, Honolulu, suggested that the people who consume cycad seed flour, this might be a link to the Chamorro disease. So we began looking at the cycads. I knew from my training in tropical biology that cycad trees have these unusual aerial roots that grow upwards through the leaf litter, and that these roots, which resemble little coral heads, actually contain cyanobacteria. So in our laboratory, we cultured these from cycad roots. Here I've cut a root with a pocket knife. You can see the cyanobacteria ring in there, and we found that they produce a very potent neurotoxin called BMAA. Let me turn the time now to Dr. Bannock. Go ahead, Sandra.
Hi, everybody. I'm Dr. Sandra Bannock, and it's a pleasure to be here with you today. One of the things that we noticed in Guam is that the people make a flour out of these cycad seeds, and it's from that white part in the middle called a gametophyte. They use it to make dumplings and tortillas. This particular material and these cycad seeds are eaten by these animals called flying foxes, which are very large fruit-eating bats. They eat these and they accumulate a toxin known as beta-methylamino L-alanine, or for short BMAA. Our work showed that they accumulated this. The interesting thing is that people eat these bats. So on the left of your screen, you see a flying fox in a bowl of coconut milk. They eat the whole thing. They eat the fur, the brains, the liver, the kidney, they eat everything. And from this, what we were able to find is that the Chamorro diet is rich in this neurotoxin called BMAA. Our work showed that the little bacteria that grow in the roots create this molecule called BMAA that then accumulates in the tree and in the animals that eat the fruit from the tree, or the cycad seeds from the tree. And that gets into the people, and then a quarter of the adults in the villages that are most traditional die of this disease called ALS-PDC. Our work showed, this is a graph showing on the left the amount of this BMAA found in Guam ALS patients. This is in brain tissue. And then we also looked at ALS patients from North America that have no link to Guam. They don't eat flying foxes, but there is exposure to cyanobacteria. And then if you look on the far right at controls, there is basically no BMAA in controls, or very, very small amounts.
So we felt that we had discovered in Guam a path to ALS. And I need to tell you that within our consortium, all of us believe that ALS is not a single disease, it's a syndrome. And that there are perhaps many different paths that lead to the same cascade of motor neuron death that we see in ALS. But we had discovered a path in Guam, but this still gave us deep concern, because we knew that you didn't have to be in Guam to be exposed to cyanobacteria. These are photosynthetic gram-negative bacteria. You've probably seen them, you just didn't know what they were looking at. Here, NASA has taken a photograph of Lake Michigan with Chicago at the bottom, Milwaukee up here, and this is a massive cyanobacterial bloom. Here's the Baltic Sea, here's Helsinki, Stockholm over here in the Stockholm archipelago. And again, a big cyanobacterial bloom. So we realized that potentially people could be exposed to this toxin anywhere in the world. And so this, although this may be a small path towards ALS suffered by the Chamorro villagers, we realized that it may not be limited to Guam. Would you go ahead here, Dr. Bennett?
So one of the things that we were able to find is that this toxin BMAA causes proteins to misfold. And what is unique among all of the diseases from ALS PDC, the ALS component, the Parkinson's component, and the Alzheimer's component, is that proteins misfold. So there's this common problem that is found in on all of the proteins that misfold. At least each of them have a protein that misfolds that is found in the brain tissue. And it looks a lot like the slinky. On the right, you see what a normal amyloid beta protein is supposed to look like. On the left is roughly what it looks like when it all misfolds and tangles and accumulates in the brain tissue of these people, which was consistent with both the diseases and what we found in ALS, the individual diseases, as well as what we found in ALS, PDC, and Guam.
Keep going.
So one of the things that we started off looking at, this is work with Dr. Rachel Dunlop and Dr. Ken Rogers. We looked at trying to understand the mechanism of protein misfolding. And what we found is that BMAA, this neurotoxin, actually interferes with the process of making proteins, and it gets inserted in the place of what is called serine, which is a normal amino acid you're supposed to have. When BMAA is present, it can actually insert itself into the protein. That changes the chemical interactions within that protein and causes it to misfold and then to accumulate within the brain.
I might also mention that Dr. Rachel Dunlop and Ken Rogers are Australians. I felt they were the best people in the world to help understand misincorporation of a non-protein amino acid like BMAA. They were working at the Australian Heart Institute. I met with them and asked them to actually stop all work on heart issues and start doing this. They agreed. And this led to their departure from the Heart Institute. They ended up in a university in Sydney. But what was fascinating with their work was that these proteins that misfold cause human neuronal cells in culture to die. They enter a programmed cell death called apoptosis. But as they added increased amounts of serine to the culture media, they could stop these neuronal cells from dying and the proteins from misfolding. We quickly went to a member of our consortium, Professor Joy Godo, who's chair of chemistry at California State Fresno and an expert on fruit flies. She tested it in fruit flies and found that BMAA gives fruit flies paralysis. When she added serine, she could stop their death and paralysis. So we thought, well, gee, maybe this amino acid could be a novel therapy for the treatment of ALS. And if you look here, we have here a drawing of what the amino acid serine looks like. This is one of 20 normal amino acids in our diet. And then there's this weird neurotoxin produced by the cyanobacteria called BMAA. And you can see that it'll be pretty easy for our cellular machinery to mistake BMAA for serine. So we found that at least in cell culture, these are human neuronal cell culture, that BMAA can be blocked from misfolding these proteins by addition of the amino acid L-serine. So we wanted to see if this could protect non-human primates. So we looked around and we traveled to the island of St. Kitts in the Caribbean. A couple hundred years ago, two vervets were released by sailors on the island. They don't belong there, but we're very interested. It says there's no predators. The number of vervets increased to about 40,000. Dr. Frank Irwin from Harvard had created a really beautiful facility in St. Kitts to study them. So we went to St. Kitts and asked if we could begin experimenting on these animals. What we really wanted to do is to see if we could produce a model of what was happening to the Guamanian villagers in these non-human primates. These are Chlorocebus sebaceous. They're socialized in groups. We were very attracted to this facility because it's like a big zoo with all the animals mingling together in groups. There's a offshore medical or veterinarian school there so that we had full-time veterinarian care. So the experiment was this. After the regular meal, we would present the vervets with a banana that had in it a powdered test substance. We dosed them orally for 140 days. And what we basically did was telescope into 140 days the lifetime BMAA exposure of a Chamorro villager, and then saw what happened. Here's our team here, part of our team in St. Kitts, where we were preparing the doses for the bananas. We had these different cohorts, a control cohort that had rice flour put in their banana, a cohort that had BMAA put in their banana, a cohort that had the amino acid serine in their banana, and a cohort that had both BMAA and serine. We were able to trigger neuropathology that the Chamorro villagers have, amyloid deposits, and for the first time in any animal model, neurofibrillary tangles. Some of you will recognize those as the neuropathological hallmarks of Alzheimer's disease. Here's our photograph. E and F is a human Alzheimer's brain. A through D are vervets that were exposed to BMAA. And then we've continued and found and published earlier this year our studies of spinal cord pathology in these same vervets. And we found that BMAA triggered the earliest possible stages of ALS in these animals, and that we're able to slow the production of that with L-serine. The lead author was a very gifted neuropathologist at the University of Miami Brain Bank, Dr. David Davis. And we conclude in the article, say we demonstrate that chronic dietary exposure to the cyanobacterial toxin BMAA causes degeneration of the upper and lower motor neurons, activates microglia. These are like the, what would you say, Sandra? Sort of like the
Brain antibodies, brain immune.
Yeah, they're T cells. They're like protector cells. And an immunoresponse activates microglia in the lateral corticospinal tracts and induces proteinopathies. That means protein deposits or deposits of misfolded proteins with reactive astrogliosis in the anterior horns of the spinal cord that are characteristic of ALS, MND. BMA dose Vervet can be used to model the neuropathology of Guam ALS PDC and ALS MND. So what we had done in St. Kitts, and we replicated this experiment twice, it was a very expensive experiment. We were able to reproduce in those vervets the same disease that we had studied in Guam, and we were able to show that L-serine reduced the neuropathology associated with ALS PDC. And here's our replication. It was highly significant. Any neurologist seeing this thinks, oh, wow, that's a human, either Chamorro patient or Alzheimer's patient. Answer, no, that's a vervet. Here's from our vervet studies on A and E. You can see these neurons. A are just normal neurons. E we've stained them for their nucleus. They're just fine. On B and F, after administering 140 days BMAA to these animals, their nuclear processes in their neurons get all messed up, and they start having these protein deposits of TDP43, ubiquitin, and other proteins associated with ALS. And here you can see on the top row is just a normal vervet spine given control. On the next two rows down, these are actually proteinopathies of TDP43. These are protein, and these are microglial responses. So the microglia are congregating here. They sense there's a problem. And the bottom row is what we see in a sporadic ALS case. So we were pretty thrilled that we were able to actually replicate this Guam disease in these vervets in St. Kitts. I wanted to see if we had any human reference, and I am an ethnobotanist. So looking around, we decided to begin studying a place called Ogimi, which is a village on the Japanese island of Okinawa. It's a matriarchal society known throughout Japan as Longevity Village. My thinking was that, our thinking was that if these people living to be over 100 years old are just so in fabulous condition, that they must have great neurological systems. And there's never been a recorded case of ALS in this village of 3,500 people. The people go out in the mornings, they collect up to 25 different species of seaweed. And what I found was extraordinary. This is a 98-year-old woman. I just filmed her here. You can see her, she has no motor neuron deficits, we can see. I asked her if she could touch her knees, and she puts her hands flat on the tatami mat. Here she goes, and bam, palms flat on the tatami mat. I want to see what this motor neuron that goes from her hip down to her ankle does. Backwards, a little stiffness, typical for somebody that's aged. But here's her legs. I asked her to lift them up. And what's impressive here is not just the legs, but the range of motion, fluidity of motion. So these people live like, or move like ballerinas. She keeps her diary in kanji characters on this little calendar. So we're very impressed with these people. So armed with these data, we went to the FDA. They allowed us to approve our application to begin a phase 1 clinical trial of L-serine for 20 ALS patients, which we began with our colleagues Todd Levine and David Saperstein, part of our consortium at Phoenix Neurological Associates. We then finished the trial with Dr. Robert Miller's team in San Francisco. We published this, and we were thrilled to see that we were able to, it was a dose ranging study. Not only was L-serine appear to be safe for ALS patients in this small study, but there was a statistical trend that we're able to slow disease progression. So that got us very excited. We went back to the FDA to approve a phase 2 trial for 50 ALS patients at the Geisel School of Medicine at Dartmouth. They approved it. We've had some problems recruiting patients to the trial because of COVID. New Hampshire basically shut their borders, and we're competing against other trials down at Harvard Mass General. But we have 30 patients now that have been enrolled. We're going to take a peek at the data. The FDA allows us to do this in the end of this year. But I want to be really clear, we do not advocate patients taking L-serine prior to FDA approval. And the reason is that we really believe that the FDA process protects people against things that can harm them. And we really want good science behind this. We don't want to sort of beat the drum here. I was thrilled when Richard Bedlock, who is one of our heroes, I think he gave us a C+ on serine on his ALS drug chart. And I called him up and said, God, that's great. I wish you would have given us a C-, because if this thing works, we will let the world know, but we don't know yet. Let me turn the time back to Dr. Bannock, because as I mentioned in the beginning, we're very interested in speeding up diagnosis. And I'd like Dr. Bannock to take a couple of moments to talk about this. And I have to say that she is an extraordinarily gifted scientist. I mean, Again, I've recruited the very top minds in the world, brought them to Jackson Hole. My model here was Los Alamos, in which weapons were created that had never existed before in World War II. Maybe we could use that isolation to do something good. So, Dr. Bannock, would you talk a little bit about this work here?
So we are really, really excited about this. And the reason we're excited is because it has the ability to change patient outcomes. What we've done is from these trials that Dr. Cox just described, we got the most amazing blood plasma, which was prepared and by the laboratories in exactly the way that we wanted. So with these blood draws, what we're looking for are things called exosomes. And exosomes are really small particles that are used to talk between cells. So you've got, one cell here, one cell over here, and they communicate with these exosomes. And what we did was we decided to pull those exosomes that came from your brain and enrich them and eliminate ones that came from other parts of your body. And within the exosomes is a cargo which contains genetic information. And what you're looking at on the screen is, in order to show that they're exosomes, there's all kinds of careful scientific things that we have to do to show that, in fact, we're looking at what we're saying we're looking at, because these are extremely small particles. They're nano-sized. And in our work, what we were able to do is to find genetic material within this cargo that differentiates ALS patients from controls, from healthy people. And these are unique signatures of early stage ALS, because that's the population that we looked at in our trial. At diagnosis, we got this blood plasma and found this unique signature. And what we're doing right now is looking for a partner to be able to make this test available for ALS patients. Anyone that's dealt with ALS, their family members, it takes sometimes up to a year, and I'm talking to an ALS community, you guys know better than I do, sometimes up to 2 years to get a diagnosis of ALS. And that's because the neurologists have to see progression of the disease over time. With this biomarker, we can look at the very first symptoms that hit and say, no, you don't have ALS, or maybe you do. And being able to make this very early diagnosis, we think will be just a real blessing for the ALS community. And so right now we're looking for a commercial partner to take this blood test and make it available. We are continuing our FDA approved clinical trials and have some things that we're looking at in the future. And one of the things that we've done a lot of work at is to reduce public exposure to these cyanobacterial blooms, which is one thing that we think can trigger the symptoms of ALS. There's probably others, but this is one, and something that's easy to do. Let's clean up our environment. There's a gene environment interaction going on here. And there are things that we can do in our lives, in our daily lives, to reduce our exposure, reduce the chances of getting ALS. And then if our clinical trials prove to be effective, we might be able to slow down progression. So if we can identify the very, very early stages of ALS, we can prevent the exposure, but we also might be able to prevent progression. And that's something that we're looking at right now.
So let me just conclude. We're in Jackson Hole. Obviously, because of COVID, we've shut our laboratory, but when the pandemic passes, we welcome visits to our lab from patients and their families. We are trying very hard to work as quickly as possible to discover new ways of slowing ALS. We think we can slow it. And we're looking at ways to rapidly diagnose it from a single blood draw. And our dream would be that you wouldn't have to get bounced around the medical community for a year, but a single blood draw could definitively tell you if you have ALS. There's some limitations to our studies. First of all, these are small sample sizes. The statistics are great, but they're still small. Secondly, we believe that we in Guam have found one path to creation of ALS. There are probably many other factors. We see cyanobacteria exposures as a risk factor. There's probably many other risk factors. And we definitely are completely wedded to the FDA process. The FDA has been very good to us. We have a very privileged relationship with the FDA. And so we really are not advocating the use of L-serine until the FDA acts. And then we think that's an important protection. So that's our presentation. What you've heard here, I would not call the mainstream research. We have a lot of people come out to Jackson Hole, and but we'll steal hubcaps if we have to off pickup trucks to fund our research. The one thing I do want you to know is we are 100% devoted here in Wyoming to getting a lick on this disease, and we're working as fast as we can, and we're pretty excited about our progress. So I'll stop there, and thank you again, Dr. Bannock. What a pleasure to have you participate in the call as well.
Thank you, both of you.
Thank you so much. That was fascinating, and you provide so much hope, and you have so much compassion. And I've seen so many of your videos and your films, and it never gets old listening to everything that you have to say. And we appreciate so much your work and your passion in what you do for those who suffer from ALS. You know, our whole team was affected by this horrible disease. And so we've lived it, even though we haven't had it, but we've seen our loved ones suffer. So anybody who has the passion and the drive that you guys show us, we are so very appreciative. So thank you for coming tonight. And we're going to open it up to Q&A really quickly. I want to let everybody know, if you want to get involved with our speech study, it's I've posted everything that I'm going to tell you now in the chat, and I'll make this quick. We have a speech study. It's going to be the largest speech study with 1,000 participants. And all you need to be is 18 or older, living in the US. It takes 10 minutes or less per week. We've had great feedback from the people that are involved. They enjoy the process. Actually, they say it's quick, it's easy, it's user friendly, and they like seeing their data shared with them. And we also collaborate with researchers. It's open platform. So we invite all researchers to be in on this, because the more people that are connected to this, the more answers that we're going to get to help others. So please join and be a part of the study. And thank you to those who are already signed up.
All right. Well, like Lisa said, thank you for coming, and we'll jump right into it. My name's Sarah. I'm one of the other Everything ALS team members, and me and Tara Tadimala will be speaking with you. So the very first question is, how does an ALS patient find out if they have elevated BMAA levels?
I'll turn that to Dr. Bannock.
At the moment, you can't. We have actually talked to some of the diagnostic companies about creating a test where you could go and do that. One of the things that we haven't told you is that there is a latency between when you're exposed and when you start showing symptoms. And because of that, we are not sure that BMAA currently in your body would actually represent things that you've eaten recently, and not the exposure that might have triggered or been a added risk factor to the disease. So we're not sure that that's a good measure. And right now there's nobody who can give you a test that's commercially available.
Thank you. The next question is, are there other locations you have found an increased cyanobacteria that may lead to BMAA exposure?
Yes, Dr. Elijah Stommel, who's a professor of medicine at Dartmouth, and his team have mapped the residences of ALS patients throughout New England. They have some really beautiful work there. They find that if you live next to a lake that's or estuary or river that's has frequent cyanobacterial blooms, that your risk of getting ALS goes up about 25-fold. Professor William Camus in France at the University of Montpellier, a very distinguished scientist, has done the same experiments and observations in the south of France in the Tau Lagoon, and also finds that there's a link between exposure to cyanobacterial concentrations and ALS. We look forward to, we're very interested here in clusters. And we look at some of the early work on clusters. These are often dismissed by scientists who are more epidemiologically trained and say, well, you know, there's just random. But when you have 5 people in the same building or 5 people along the same road who have ALS, we think that's not a random occurrence. So I should emphasize that our focus as a team, although our diagnostic is based on microRNA, our focus is on environmental exposures. Most other researchers are working on genetic factors. And that's because we can help prevent environmental exposures. We think that's a pretty easy and straightforward thing to do. Well, I'll stop there, sorry.
Thank you.
Are there any other types of food that may include the same cyanobacteria and BMAA as the fruit that the bats were consuming?
I'll toss that to Dr. Bannock.
That's a really good question. Don't eat fruit bats. That would be my first suggestion. Really bad idea, but there's some really good work that's been done both in the Baltic Sea and in Florida to show that there are marine organisms that, or freshwater as well, that can contain BMAA. And it appears that it's really high in, if these organisms are growing in the water below cyanobacterial blooms, then they can accumulate this toxin. And things like that we found high in areas that had high blooms would be pink shrimp, blue crabs, but you know, again, only those that were that lived in waters that had high concentrations of cyanobacterial blooms. So it's really hard to tell. You can't go to the grocery store and say, oh, I can't eat shrimp, because your shrimp might be just fine. It just depends on where they're sourced. And so one thing that we could do is develop some, you know, some consumer advocacy that we know where these things came from. These could easily be tested before they get to grocery stores to see if they contain BMAA. But right now, it tends to be marine organisms, clams, oysters. We found high concentrations in areas like Louisiana, that in places where there are blooms, we can see the concentration of the BMAA in those organisms. And I'll stop there.
Thank you. Kind of going off that question, how do people know where potential cyanobacteria blooms may currently be occurring? Can this information be found online right now?
It depends what country you live in. The European Union has very strong reporting requirements and legislation against cyanobacterial toxins. The United States varies by state. The EPA has not, they've come out very aggressively against our research, which puzzles us, because they would be the organization that you know, should protect people. Cyanobacteria in a bloom, there's cyanobacteria everywhere in the world, but when they bloom because of pollution or warmth in a lake or estuary, it produces a big mat of sort of blue-green gunk. Looks like a mat of scum. If you see that, get away. Do not be around that. We've focused recently a lot on Florida, where the Corps of Engineers has released nutrient-laden water from Lake Okeechobee down the St. Lucie River and down the Caloosahatchee River. Their goal is to make sure the lake level does not rise too high, but this has caused havoc with cyanobacterial blooms exploding in the St. Lucie watershed down to Stewart, Florida, and in the Caloosahatchee into Port Charlotte, Fort Myers area. And we're deeply concerned about this and are, again, working very hard to try to get accurate information that comes out to people. So if you know where a bloom is, you just avoid it and don't go there.
you believe exposure to BMAA is a one-time dose or a potential long-term exposure like you saw in the Guam population?
That's an excellent question. Our experiments and our studies are on chronic exposure. And it may be that it's the chronicity of insult, the frequency of insult more than the dose that triggers the response. But some of our colleagues, Carlson and others in Uppsala University in Sweden, Tim Downing in South Africa, have found that neonatal populations, in other words, if a mother is exposed, and this is in mouse models, that her offspring have a higher likelihood of motor neuron dysfunction when they're adults. Clive Sable at the University of Exeter, who on our consortium has studied in Finland. It's difficult to do this work in the United States because of privacy, patient privacy laws. But in Finland, they don't care about patient privacy. They just want to cure the disease. So they'll tell you where any ALS patient was born and died. And statistical analysis of that shows that if you were born near a big lake that has cyanobacterial blooms, that your chances of getting ALS down the road as an adult really increase. So it may be both. It may be both chronicity of exposure. It may be there are certain parts of our life cycle where we're very vulnerable to it.
Thank you. The next question, is the only way for a person to have BMAA in their system be through ingestion, or can it also be absorbed through the skin, as in through swimming?
That's an excellent question. The answer is we're not sure. We have recorded, we have a paper on an ALS patient in Florida who would go to Florida Bay and eat lobsters, and we tested these lobsters out of his freezer. They're just chock full of BMAA. We did the same thing in Annapolis, where unrelated people on the same street would come down to ALS. They're eating oysters that had it. So I want to come back to the question. I think I've missed the point of the question. Could you repeat it, please?
Yeah, no problem. Is the only way for a person to have BMAA in their system be through ingestion, or can it also be absorbed through the skin through other avenues such as swimming?
We don't know about skin absorption. It is a small molecule. We think ingestion is obviously a route that needs to be looked at, particularly people that eat oysters, mussels, other shellfish that absorb the molecule when they're in a cyanobacterial bloom. But something that's very disturbing to us is the possibility that it's aerosolized, that you could breathe it into your lungs. And we're engaged in work now with Professor Stommel and his team at the Geisel School of Medicine at Dartmouth University. We're actually looking at lung tissue of ALS patients, looking for cyanobacteria and cyanobacterial products. So stay tuned. We don't know yet. We do know that it can aerosolize, and this is work done by some folks in Florida and Stommel's team at Dartmouth, but we're not sure yet.
Lots of interesting information going to be coming our way soon. Do you believe that maybe BMAA exposure could trigger genetic versions of ALS, cause a mutation that, or show one that has already been inherited?
Let me toss that to Professor Bannock.
We don't have really good data on that. There is some work. There are people that have the SOD1 mutation, the Sami people up in northern Sweden, who do not come down with ALS. So it is certainly possible to have genetic risk factors that, and they don't produce the disease. So what we think now is that it's a model of risk factors. And so just like cancer, where you have to have repeated risk factors or a certain number of risk factors in order to get the disease, we think that's what's going on with ALS, is that BMAA is one risk factor, genetics is a risk factor, age is a risk factor, gender is a risk factor, and there may be other environmental insults that are also risk factors. And if you add up enough of those, then you can cause enough insult to your neurological system to create the disease. But there's still a lot more work that needs to be done on this.
Thank you. Would the exosome test work to determine if family members have ALS? Is it progressive monitoring, and how often would the test have to be administered?
These are great questions. One thing we're excited about is that, and we're in discussions right now with a diagnostic firm to try and make this available to patients and their families. Not only could it lead to rapid diagnosis for ALS patients, but it could also comfort family members who have a negative result from the test. How long it'd have to be repeated, how frequently, we don't know. Our hope is that we could actually use it as a predictive test of ALS risk. But we're moving forward. My dream, frankly, would be that everybody that reaches their 40s or 50s, just during a regular doctor's visit, the doctor clicks off on a little piece of paper this test, and most people no worry, but there's a couple that, 2 per 100,000 that they're brought back in. The one thing that's exciting about this is if we can actually push this back into the pre-symptomatic stage, this would allow earlier treatment. And it may be that some of the drugs that have failed in previous drug trials to treat ALS will be far more effective in an earlier stage. So we're pretty excited about this diagnostic test and are working as hard as we can to get this out to the ALS community.
Wonderful. In your presentation, you spoke about L-serine. Is there a dietary mechanism in order to gain more serine for people with ALS?
Right. And again, remember that we are bound by FDA license, and so we are not encouraging anybody to take L-serine. We suggest that people consult with their own physicians and neurologists and follow their advice. Because of our studies in Japan of populations that have no record of ALS, we find they get about 3 to 4 times the amount of L-serine in their diet that North Americans do. Sources of L-serine on our diet include edamame or other soy products, pork, turkey, sweet potatoes. So now, when I go to get a hamburger, I always ask for sweet potato fries. So it is possible, and seaweed. So that's how they really get a lot of serine in their diet. So you can tweak your own diet, but again, maybe we're getting ahead of the horse here. The cart ahead of the horse. I'd like to get the results from the clinical trial to make sure that it really is working, that we can replicate what we found in the phase 1 trial. If not, we'll drop it like a hot potato and move on to the next possible treatment. But at this point, we're very invested in trying to see if L-serine works.
Great, thank you. In the same note, for the study, how are you sourcing the L-serine? What is the dose of L-serine and how long is each patient taking it in the trial?
All of one of the joys of being a not-for-profit organization, we don't have secrets. You can go on to clinicaltrials.gov, and our clinical trials are listed there, and you can plug in L-serine and see what we're doing. Currently in the Dartmouth trial, each patient is given 30 grams of L-serine per day. We used our own pharmaceutical manufacturer to prepare those doses, so we don't have any advice. Either we do not advise people to take it, and we definitely do not endorse any commercial products. And this has been a problem with this because we've had in the last 2 months 3 or 4 websites suddenly showing our image, showing our research, and pushing L-serine. And you need to know we haven't given our consent or permission for any of that. We are not advocating the use of L-serine. We're a not-for-profit organization. We have our sole interest is helping ALS patients. That's it. We don't have any other interest. I mean, if I want to make a bunch of money, I'll go to Wall Street. Well, maybe not after today, but anyway, this is not, all of us are deeply, deeply devoted to improving patient outcomes. We want to make sure the information we give the ALS patient community is accurate, that it represents replicated science. So we're excited. We think we're there at the diagnostic test with serine as a possible treatment to slow ALS. We won't know until we finish the clinical trial.
Thank you for that. Are you, in terms of just how L-serine works on the body, I know you showed the amino acid itself, but how is it different than maybe another amino acid like an L-lysine, and what does that difference mean for patients and their proteins?
Professor Bannock.
Every amino acid that's normally in our diet is connected into proteins like cars on a train. One's connected to the next, and there are 20 normal amino acids that are there. Each one is important to make a protein. You have to have every amino acid in the right order to make that protein. So they're all very, very important and very different. Some are called essential amino acids. that we have to get from our diet. Others are called non-essential amino acid because our bodies are able to make them. But you know, each one is a little different and higher in some foods than others, but basically all 20 are needed for you to function.
Great, thank you. The next question, could other things be replacing the L-serine in proteins other than BMAA?
The answer is very, very possibly. There's 20 different amino acids, coming back to Dr. Bannock's analogy of train cars all linked together. There's 20 different types of those cars in our body, but there's over 1,500 non-protein amino acids. And we've been very interested in our laboratory in the misincorporation of those. L-serine, though, seems to be somewhat special because we initially thought, well, great, it was a protection against BMAA exposure, but again, BMAA exposure may be a very small, minor path, an uncommon path to development of ALS. But now we've been doing work, and this was really pioneered by a very gifted molecular biologist, Dr. Rachel Dunlop, who we actually stole out of Australia and kidnapped her, and she's in Jackson Hole. We're very grateful for Rachel. She's found that L-serine alerts what's called the unfolded protein response in our mitochondria and in an organelle called the ER. So it's base endoplasmic reticulum. So it's basically tells the cells, watch out for proteins that are misfolded. It quickly deals with those. So it sort of gives a heightened sense, almost like maybe an immunity against many other types of protein misfolds. So that was a very good question. I'm grateful for it. Thank you.
Do you think that ALS patients should eat the seaweed like the Japanese population people do that you studied? And if so, what kinds were they frequently having in their diet?
Dr. Bannock.
You can't get a lot of the seaweeds that they were eating here in the Western world. They're eating a lot of very unique things that are there. I mean, it won't hurt you to eat, certain kinds of green algae that we know of, at least, but we don't know. You can't just go to the grocery store and buy what they're eating.
Well, some of these folks may live in places where you could buy, Asian foods, and we've been impressed with a couple of seaweeds, of course, kombu in Japan and others. But again, I think one of the highest sources of L-serine is edamame, which now I eat instead of popcorn when I'm watching Netflix. So.
Thank you. The next question, are you continuing to recruit for the trial that you're doing this year? And could patients from other centers be a part of that trial?
The answer is yes. We have a license from the FDA for 50 ALS patients. We're at 30. It requires 3 visits to Dartmouth. Blood samples are taken. You're very carefully observed. And the thing that we've been facing recently is that New Hampshire basically shut their borders to other states, but that's opening up hopefully. And if you're interested in that, contact us. My email is paul@ethnobotanists.org. We will put you onto the clinicians at Dartmouth who make the decisions of who are allowed in the trial. But typically it's, I think the criteria are diagnosis within the last 3 years, functional vital capacity over 60%, and a certain minimum ALS-FRS score. I'll tell you, I've been so impressed with the Dartmouth team because they really care about patients. And that was very important to us when we chose a site to fund and sponsor the trial. I really hope that Dr. Elijah Stommel wins a Nobel Prize because I watch the tremendous compassion he has for patients. He brings his cello and plays it for patients. You get house calls from this guy. I mean, he's terrific. So yeah, and you could find the details of this trial on clinicaltrials.gov. If you put in ALS, you'll see all the ALS trials. If you put in L-serine, you'll see our trial.
I guess somebody was very active and was able to go to that site. And their question is, why was the 30-gram dose chosen?
The answer is the 30-gram dose was in our dose ranging trial, the phase 1 trial, showed that the greatest slowing of disease progression is measured by the ALS-FRS score. And that was a very small trial, but we had an 85% slowing of disease progression. So we decided just to put everybody on 30 grams a day in the trial. What we want to see is if we can replicate that big effect in the Dartmouth trial. If we do, then boy, we're going to beat the drum and let everybody know. But at this point, we're not ready because we don't have the data.
Thank you. Can spirulina expose you to BMAA? And if so, what are your thoughts on this?
Dr. Bannock.
Yes, there are cases where spirulina has been tested and shown to contain BMAA, small amounts. There's also been spirulina that's been tested where BMAA was not found. We certainly want to be cautious about eating cyanobacteria directly. What we're finding is that BMAA through magnification through food webs gives you higher concentrations, but it is certainly something that's on our radar and something that we should think about.
Right. Kind of go back, Dr. Cox, to what you were just saying. You were talking about how it stopped nerve degeneration. Do you expect any reversal of nerve degeneration by the L-serine, or just arresting of that degeneration?
You know, reversal, of course, is the holy grail. And there are people walking around and reversed. I think, again, Professor Bedlack, one of our heroes, has been interviewing these people and compiling a list of them. So there's also some people that plateau and stop progression. I do not understand the mechanisms of that reversal. I doubt they have anything to do with serine. I seriously do. But you know, we'll take whatever we can get. What we really want to do is to buy time for patients. And I think at this point, a realistic goal for us is to change the landscape of ALS so it becomes a chronic disease that's managed, much like multiple sclerosis, and it doesn't become a killer. And we really want to buy time for patients. To me, that's the most important thing right now, is to increase lifespan, increase quality of life. And so you can see we're doing two things here by coming up with a new microRNA diagnostic test that we can get a faster diagnosis so people can get on the treatment. And secondly, trying our drug, and hopefully people will use our diagnostic test to test other ALS drugs, including some that have been rejected previously. So our focus in Wyoming right now is to change the landscape so we buy time for ALS patients and their loved ones and improve the quality of life.
Great, thank you. And our last question is, are veterans more exposed to BMAA?
We noted that there was a tripling of, epidemiologists found that there was a tripling of ALS among personnel deployed to the Gulf in the first 1992 Iraq conflict. And that was a very careful study. It was compared to people that had received the same training but had not been deployed. So I took Dr. Walter Bradley, who is arguably the top ALS neurologist on planet Earth. This guy's like Yoda. I mean, you feel a tremor in the force when you're around Bradley. So I said, why don't you come with me to the Persian Gulf? So we went down to the Arabian Gulf, I should say, and we freewheeled around Oman and Dubai. And of course, the major reason was that this could disprove our hypothesis, because most of the time we think about cyanobacteria, we think they're waterborne. In Qatar, the soil looked really funny. It's a flat lake like in Iraq and Kuwait, old lake bed. Poured my water bottle. It was August, really, really hot. It was probably only 104 degrees, but the story's so much better if I said it was 120 degrees. It was really hot. Poured my water bottle on, boom, the cyanobacteria start reproducing. They start greening up. With our colleagues there, Aspa and Renee Richer at Cornell Weill, medical school, we began studying intensively the Gulf region. And we believe what happened is that in that deployment, it happened so quickly, a lot of American soldiers and others did not get warm clothing. People forget that it gets cold down in the Gulf during January and February. They were walking behind tanks and trucks to try stay warm. And we think these trucks and tanks were aerosolizing this dried cyanobacteria. We found, our colleagues found that about 87% of the state of Qatar, and I imagine it's similar for Iraq, are covered by cyanobacteria blooms, are just dried, waiting for these 2 or 3 weeks of rain in February, in which they do their whole life cycle. We've also published some recent papers where we've actually excavated pits down into the desert, looking at these cyanobacteria, which have been there for centuries, pumping out toxins. I do have to say that we've been treated so remarkably kindly in the Middle East. It's amazing when villagers find out what we're doing. Even though I have very limited Arabic, we've been treated with great kindness and compassion by these people. And so we're very concerned about our veterans. There was a time-related spike within about 10 years of exposure. Boom, here's this spike. And we've been trying to work with the Department of Defense to encourage that soldiers are protected.
Okay.
Dr. Bennett, did you want to add anything to that last answer?
That was great.
Okay, I think that's it for questioning. If your question did not get answered, we'll send all the answers to the questions and additional questions. Hopefully, Dr. Cox, if we can send them your way, and Dr. Bennett, and get answers to everybody if they feel like their question wasn't answered. So again, thank you so much for your time. And we look forward to hearing great results from your clinical trials. And I'm hoping that your diagnostic tests can be commercialized soon. So
we're very hopeful. And Lisa, I just mentioned that people can look at our website, brainchemistrylabs.org, and get more information. But we're not for profit. We work for you. We work for this patient community. You are our bosses. We're trying very hard. We're doing very different sort of research here in Wyoming that's being done elsewhere. But so far it seems to be promising, and we're excited to keep going. The website again is brainchemistrylabs.org.
allow visitors to have a tour of your facilities?
We absolutely do. We closed the labs due to COVID, and we've all been quarantined because we think the work's too important to slow down. And unlike many labs, we did not pivot towards COVID. We just kept it out of the lab. But once that's abated, we would welcome anybody. And look, if you make your way to Jackson Hole and you're a patient or patient family, if I have a sandwich, I'm going to give you half of it.
Oh.
I really care about it. And we're going to make your visit as wonderful as we possibly can. And every one of my scientists have that same commitment. So of course we welcome you. But at this point, because of COVID, we have to have no visitors. Hopefully that'll change by next year.
Thank you, Dr. Cox. And I'm one of the founders of Everything ALS, and I just wanted to let you know our organization is very focused on diagnostics and finding ALS earlier. So we would love to talk to you more about your diagnostic tests, and that's one of the reasons why we're doing study of speech as well, to come up with early diagnostic tools and also tools for clinical endpoints.
Excellent. Well, thank you very much, everybody, for being with us.
Thank you.
Thank you so much.
Thanks
Have a
for
good
having
evening,
us.
everyone.
Yeah.
Well, everyone, welcome to another adventure on Everything ALS, brought to your living room.